Okay, the last subtopic of chapter 4 reproduction is this one, growth in multicellular organisms. Now, growth phase. Growth means a series of change that occurs in any organism starting from the zygote until to an adult. Now, apart from growth, there is something called development also occurs. Now, growth has different definition, and the most immense one, and according to textbook, is growth is something irreversible. It's supposed to be a permanent increase, and you can measure through the following parameters: size, length, volume, height, surface area, cytoplasmic mass, dry mass, and number of cells. It is very important to know the definitions of growth because it occurs mostly in paper 1 questions. Now, definition of development means cell differentiation. Apart from growing, right, sometimes some cells will differentiate to become unique cells, and the differentiations can be a qualitative change that you can observe in the form of shape, how it specifically becomes into something else, and the complexity as well. Growth phases involve three things: the cell division, the cell starts to divide through mitosis, the cell elongates, the lengthening of the cells, and then some cells differentiate to become some unique cells. Three different phases: division, elongation, and differentiation. Now, phase 1 is cell division. If you refer to the diagram over here, look at here. There's one. Alright. Now, after telophase, during telophase, the cells will divide into two. The 2 becomes 4, 4 becomes 8. It can be in this form where you can see it horizontally, or it can be in a stacked up form like so. But still, the mitotic division occurs, and the mitotic division occurs in all somatic cells. Somatic cells means all the cells except for the reproductive cells like sperms or um, ovum. Now, cell division in plants mostly occurs in the apical meristem. Now, apical meristem is a structure that actually you can find in the root tips or the shoot tips. Shoot tips means at the peak of the shoot, at the beginning of the shoot, and root tips means at the very beginning of the roots, the head part of it. Now, if you look at the apical meristem over there, the cells are quite small. It's closely arranged. They have large nucleus. They have a very thin cell wall because they are actively dividing, but they don't have a vacuole because at this point the cells are actively dividing, and vacuoles here means they have to act as a storage. Okay, so any actively dividing cells, which means like they are in the verge of like continuously doing some stuff, they don't have the time to do a storage or something. Hence, this makes them to have a dense cytoplasm. So that means the body is covered by whole number of cytoplasm. Now, after the cells have divided, right, since the earlier part over here, the cells are very small and everything, at one point the cells will grow. Grow means they will become longer and longer, they will elongate because the vacuole will be either they don't have it or they have started to develop it, or they have already have it but in a smaller size. Though, so cell elongations is only possible when the vacuole actually absorbs water. So the vacuoles also increase in the size, and the small vacuoles will become a large central vacuole. When the vacuoles collect all the waters as shown in the diagram here, the cells become bigger, and they become bigger in sense that horizontal cell elongation occurs. Now, next thing, some cells will undergo differentiation because they have to perform some special functions. So as in the diagram, you can see neurons, epithelial cells, sperm cells, ovum cells are called specialized cells because their cells does not only perform normal function but they have a special functions. But you can see in the change of shape and the specificity. As for plants, right, meristematic vascular cells, which is xylem and phloem, are called specialized cells. As for humans, we have a lot like neurons, epithelial, sperm, ovum, and so on. But for plants, we can merely say they have specialized to become xylem and phloem, the main one. So this is from the apical meristem, so which has become meristematic cells. Now, cell differentiation in plants also apart from xylem and phloem, you can find the gut cell, the mesophyll cell, palisade mesophyll, spongy mesophyll, and also the parenchyma cells on the cortex of some tissues and something. So there are five different cells that are specialized for plants. As for humans and animals, there are a lot. Now let's look at how growth phases occurs in plants in a root tip. If you look at the root, right, as I told you, this is the root cap over here. The root cap, here you can find abundance of apical meristem root cells actually. So apical meristem's root cells are found abundantly in here. So which means cell division will occur rapidly over here. When you go to the next stage, the second part over here, cell elongation will happen rapidly as well because this is where the vacuoles has been developed, or maybe earlier they have a very small vacuole and it will start to collect all the waters and the waters will enable the lengthening of the cells. And the third one is the zone of maturations. All the cells and most of the cells will actually undergo maturations, which means they will mature to become a specialized cells such as the epidermis root hair, vascular cylinders such as xylem and phloem, endodermis, pericycle, and so on. So upper part, all they have become some specialized stuff. Now let's look at the plant shoot. Plant shoot also has the same thing. On the very peak over here, you can find abundance of apical meristem shoot cells. So they are actively dividing. The next stage is the cell elongation. So they have vacuoles to collect all the waters. And the third part, you can find cell differentiation, the same thing. You can find xylem, phloem, cambium, leaf, shoots, and so on. Okay, now how to measure the growth? This is the second part of the learning. Since you know what is growth, growth is irreversible permanent thing that you can actually measure quantitatively in the form of several parameters. Okay, some growth are also qualitative. You can see in the differentiation in the shape, specificity, and the complexity as well. But it has to be irreversible and permanent. How to measure growth? Now, measuring growth, first one is surface area. You can also take surface area as the parameter. Very simple. You take a leaf or any structure that you would like to determine the surface area, put it on the graph paper or square paper and count how many squares are inside. It's supposed to cover 75% of the squares. So an example is given over here. Okay, now, there are other parameters as well for multicellular organisms, such as the weight or the mass. We have two different weight or mass, which is called the fresh and the dry one. The next one is the length or height, which is very popular as well to predict the linear measurement. Okay, let's talk about the fresh mass. Now, fresh mass means you are taking the mass of the organism as is it, for example, in this normal condition. Okay, now the advantage is like you are how you are right now, like without actually taking any part of your body, like try to evaporate water or what. No, just normal condition, just take it as the way you are. So the advantage is it's easy and convenient. You don't have to destroy the specimen, or you don't have to remove anything like water content or something. And of course, in this normal condition, you can use back the same organism because you did not destroy it for another repeated measurement. Now, disadvantage is it can be inaccurate and inconsistent. Why? Because, well, every organisms have different fluctuations of quantity of water. You see, as we know that our body mass is 70% roughly around that, composed of water, so it does not give a real parameter of how the growth has occurred. The growth may be what you're considering is just the 70% of the water. Now, dry mass, on the contrary, can be a great curve for that. If you want to see the real growth in terms of mass of the organism after you eliminate the water content, so it will show the actual growth. However, the minus point, the specimen is destroyed. And these are the methods. To do it, actually, you have to destroy the specimen, transfer it into the oven at 110 degrees Celsius to eliminate all the water, and then you weigh it, and you weigh it until you get a repeated constant value. So advantage, more accurate because you eliminate the water. Disadvantage, you're destroying the specimen. And of course, you can't do a repeated measurement because the specimen has been destroyed. You have to use another one. Now, large samples of genetically identical organisms, you have to grow it in a similar condition. Likewise, I said, if you take one species of a one kind and you want to do a repetition, it's not possible. And this process is also time consuming. Now, let's look at the growth curve right now. Now, growth curve, some certain organisms follow a sigmoid curve graph, like so in this picture. As you can see, the graph shows very slow growth, rapid, and then a stationary, and then a decline growth, which means there's no more further growth here, zero. Let's look at which organism has it. Now, sigmoid curve has few different phases. The lag phase, the log phase, which is very rapid, stationary phase, and also the decline phase, which actually resembles senescence and death. Now, at the lagging phase or lag phase, the growth process is very slow. Now, cell division might be very slow because there's not enough nutrients or energy to carry out the metabolism. And you can relate it when you were very small, when you're very infant, when you don't have that much of growth. Actually, you are actually experiencing slow growth from the age of 6 to 9 or 10 in that moment. Now, exponential growth. Exponential growth, or the log phase, is the phase where you grow rapidly. You can actually see the height of yours after you are in the age nearing puberty or after you have reached puberty. You'll grow taller and very rapidly. This is because mitosis happens rapidly. They have enough water to cause cell elongation and also a rapid cell division because of nutrients and energy. Now, maturity stage is the stage just right before the stationary stage. This is where the growth rate will become slightly slower and indicating that you might not have enough growth after that. Okay, so in this case, the cell division will happen slowly, and stationary stage is a flat stage where the growth rate is zero. Cells are still dividing in order to replace the damaged or dead cells, which means if one dies, another one actually will grow to replace only one. There will be no like multiple division in order to replace one to cause growth. Now, senescence stage or the decline phase, this is growth rate negative because you will lose a lot of tissues due to aging process. The cells also will die, and this is indicating the death of organism, which is nearing to that. Now, humans growth curve, there's no video. I will show you another slide to go to that. And this is the human life cycle. From when fertilized egg, fetus, to an infant, child, teenager, adult, old age, and to death. Okay, this is the growth curve. Childhood phase. Okay, infant phase is the lag phase. Childhood phase, as you can see, is the log phase. Adolescence phase is slowly like maturity phase. And yes, adult phase is the stationary, and decline phase or the senescence phase is the aging phase. Okay, at birth, rapid growth, slow growth, very rapid, very little, and death. This is very important. Okay, for growth of a boy and girl, there's a difference as well. As okay, the lag phase for girls will occur earlier compared to boy, because girls will reach puberty earlier than boys. As you can see, girls grow at the lag phase earlier than the boy. Now let's look at growth curve of insects. Now insects have several growth phases which are called intermittent. Intermittent, they actually grow, stop, and grow, stop. You can't find a sigmoid curve for that. Okay, let's look at the life cycle first. Every insect starts from eggs, the eggs being hatched, and after being hatched, the insect will undergo first stage nymph. First stage nymph means they are still growing. They grow to a point until the exoskeleton, the waxy layer on top of their body, becomes harder, and it's time to do a molding. Molding means they have to remove the skin. Okay, and after they have removed the skin, they will go for the second stage of nymph, which means they have a very fresh cells, very fresh skin which is not hard yet, and that will allow the cell division to occur rapidly, and until the cells has outer layer of cells has become harder, they have to go for the second molding. And the molding and the growth process happen like 5 stages, 5 times, until it becomes an adult. Adult means there will be no more growth. Okay, now the life stages here, they have divided into few instars. You will get to know later. Okay, first instar, second instar, third, fourth, fifth, and the last one is adult. So they mold their skins 5 times. So this is the graph, a staircase shaped graph, intermittent growth. Okay, now as you can see here, from this point, which is a very flat line over here, is the line where it shows the eggs has been hatched and it was time like they were still in the baby, the egg. And the eggs hatch, so you can see a vertical line going upwards. This is the time when the first nymph or the first instar occurs, um, whereby the insects will actually start to grow. The cells will grow inside their body until at one point the exoskeleton become harder. When you see the horizontal line here, the exoskeleton has become hardened. So it's time to do the shading of the skin, which is called ecdysis. Shading of the skin. So shading of the skin occurs at this time. So that's why the graph is flat. That means there is no growth. And after the skin has been removed, so there's a chances for them to grow, remove, grow, remove. And this occurs 5 times until the adult process is the last stage. Okay. So the first instar is on the horizontal line here, second instar, third, fourth, and adult, as you can see. Okay. Now let's look at plants, the primary and secondary growth in plants. Now, plants growth, you can actually divide them into two categories, the length and the width. Plants also grow horizontally and vertically. Okay, let's look at the vertical growth. Vertical growth, I have actually explained to you the apical meristem earlier, so but let's look at detail in a moment. And the horizontal growth means how it grows in size in a horizontal way, like the lateral meristem. We have apical meristem on the vertical and lateral meristem on the horizontal. Now, meristem tissues are dividing tissues. You can categorize into two. Apical meristem you can find on the vertical, root and shoots, tips of roots and shoots. They increase its length. Lateral means the cambium and the cork cambium, which you can find in the increase in the horizontal growth. Now, all plants will exhibit primary growth, which means all of them will show the increase in the height and the length. But only woody plants, please take note, only woody plants will show secondary growth, which means a growth in the horizontal. Okay, herbaceous plants will only show primary growth, grow in the length and the height. Now, primary growth, as I have explained earlier as well. So it leads to an increase in the length. Length means the height. So apical meristem, shoot and root, they undergo cell division, cell elongations, and also cell differentiation. Okay, and this one has been explained earlier as well. Now, secondary growth in plants for woody plants is the thickness of the plants. Now, this is due to the lateral meristems. Okay, as you can see, some plants are very big that you can't really find the diameter is too big. Okay, how this thing actually happens? Let's look at the structure of dicotyledon stem. This is a stem part. As usual, we can see the red color section here is the xylem. Xylem, and above one is the phloem. And in between the xylem and phloem, there is one section which is called cambium. Okay, cambium, and the green color things is called cortex and epidermis layer. Now, what happens here is that the vascular cambium and the cork cambium will actually grow and divide. Okay, look at this picture. The cambium ring here, the cambium, the vascular cambium. The vascular cambium will divide and then repetitively divide over here. So it will separate the phloem and xylem further actually by dividing. So when they separate actually, when they divide the cells, right, the cells are known as the primary and the secondary phloem, and primary and secondary xylem as well, as you can see. So this thing happens repetitively until they form like an array of few secondary xylems and few secondary phloems packed together. And on the other side, there is something called cork cambium as well. They divide outside and they form cork outside actually. Okay, this is the picture over here. Now this is the vascular bundle. You have primary xylem and primary phloem outside, and when they divide, as you can see, we do have primary, but on top of that we have secondary, then cambium, followed by secondary phloem, and then outer one will be primary phloem. This is how it looks like. So secondary xylems here, primary xylems in the very middle. There is a cork cambium here, and then as you can see, the yellow color line over here is secondary phloem, and then followed by primary phloem. Now each year a new ring of xylem is produced. Okay, and yes, for seasonal plants, right, where they follow spring and autumn and things, so the growth will be accordingly, like when they have a fast growth and slow growth accordingly. Now this is the root part. Xylem is over here in the middle, and the phloem is here, and the cambium is over here. So let's look how they divide. The same way they divide, as you can see, I have primary phloem 4 over here and the xylem in the middle. The xylem divide to become, the cambium ring divides to form a secondary xylem over here, and the primary is over here. Secondary phloem and primary, the small 4 dot buttons, and this one repeatedly goes on until you can see this structure. So the chalk cambium is over here. Okay, so that's all, that's the end, and this is just a review of that. So you have learned through the definition and the 3 phases of growth, how to measure growth, dry mass and the fresh mass, and the growth curve of sigmoid phase in humans, in insects, and is also secondary growth in plants and primary growth in plants. A few pictures for your reference. Okay, so that's it, and I will also show you this one over here. Again, so exoskeleton, there are a few stages. So insects have hard chitin layer which enables them not to grow, and this is the graph here. Stage 5 to stage 5 is the nymph, or what we call as instars. Okay, shading of the shell or chitin is called epididymis. The skin is being removed. So at this point, at the red color structure over here, all are growth phases. Okay, growth happens, and after ecdysis, new skin is still soft. That means cell division will occur rapidly. And at these stages, there will be no growth. So there will be no growth and it's called instar. The skin is hard and ready for ecdysis, which means molding. Okay. Now, as you can see in picture here, on the right hand side is the new skin has been developed. That's the real insect. On the left hand side is the old skin. The animal will suck in the air to expand its body first. This is how they remove their skin because they need some air to get detached. Old exoskeleton will then break, and after that, insects with new exoskeleton will then grow. Right? So animals will eat a lot to build tissue and during this time because at the NIM stages, and hormones also controls the ecdysis process. So humans one is like so, the graph, it's slightly like sigmoid as well. And the woody plants. Woody plants, you can know because every year the cambium actually divides, right? It produces secondary phloem and secondary xylem. You can actually know the age of the woody plants by looking at how many layers of that actually. So when you see this is two layers over here, the first, like if you have one circle over here, this is first year, second, third, fourth, fifth, and so on, and then actually 24. So you can know by this age. Okay, so that's it actually for the last subtopic.